David starting?
Good morning.
David Ting.
My name is David Ting. I'm the branch chief that oversees this project. I hope everyone has a copy of our agenda, and as you can see, we have a packed schedule, and we are going to get into some of the design details of some major components of our study. But before we do that, in this session, we would like to give you an overview of what is going on in this country as well as overseas. And after that, Dr. Patty Wong is going to give you an update of our project. So in this country, US EPA has sampled 40 fields and in 4 census regions, including 21 community fields and 11 military fields. And they're going to study, chemicals in crumb rubber on playgrounds and playing fields. They are going to analyze, or they have analyzed particle size distribution of field samples, and they are conducting chemical analysis using emission chamber, solvent extraction, and biofluid extraction. Consumer Product Safety Commission is also planning to conduct a national survey of children's behavior on playgrounds and identify exposure factors. National Toxicology Program is conducting short-term toxicity studies on crumb rubber in response to a request from OEHHA. They are going to conduct in vitro self-based tests and also in vivo animal studies. It will expose test animals to crumb rubber via diet, gavage, and bedding. In addition, the National Institute for Occupational Safety and Health is exploring the feasibility of studying worker exposure. Now, in Europe, they completed 2 studies recently. The first one is by the European Chemicals Agency. They conducted a risk assessment using published monitoring data, and they looked at over 100 synthetic turf fields from 10 member states. And they investigated many chemical groups, including polyaromatic hydrocarbons, pHs, phthalates, metals, formaldehyde, benzothiazoles, methyl isobutyl ketone, and benzene. And they issued their report in February 2017, and they concluded that there's low concern of lifetime cancer risk for players and workers for pHs. And there's negligible concern for players and workers regarding metal migration. And they also concluded that there's no concern for players and workers for phthalates, benzothiazoles, and methyl isoketone. Another study was conducted by the National Institute for Public Health and the Environment. They evaluated health risks associated with rubber granules, and they sampled over 100 synthetic turf fields in the Netherlands. And they also looked at more or less the same group of chemicals. And they conducted migration tests in artificial sweat and gastrointestinal juices, and also evaporated air. And they released their report in December 2016. In that report, they concluded that health risk is negligible, and there's no indication of a link to leukemia and lymphoma. Dr. Patty Wong will give you an update of our study.
Copy of the slide is in your binder right after the agenda. Good morning. This presentation provides an overview of some of the ongoing activity of our study. especially during the last year since we met in February 2016. Our synthetic turf study consists of Our study consists of 7 tasks, so I'm going to go through each task and give you the progress. For the first task is expert panel and interagency consultation, and OEHHA has been in frequent contacts with scientists in federal EPA, Environmental Protection Agency, ATSDR, Agency for Toxic Substance and Disease Registry, and NTP, National Toxicology Program. And we also participate in bi-monthly meeting with all the states to communicate and exchange information. And we have been in communication with the European Chemical Agency on their studies. Task 2 is the hazard identification. OEHHA conduct literature search on turf-related study and to identify information that can assist in chemical identification. This will be discussed in more detail in a later section. Task 3 is on exposure scenario. OEHHA invite Dr. Diana Cassidy from University of California, Davis, and she conducted a pilot study for us last year and submitted a report in May 2016. In the study, she did survey soccer player and football player, and she provide information on time activity behavior and finalize with high and low exposure for the player, and also submit a recommendation on how to develop a full-scale time activity study for our turf study. Task 4 focused on chemical analysis of sample. We have been working with the Lawrence Berkeley National Lab, our contract lab for this study, and they are here to present some of the preliminary data. We are in the process of developing procedure for analyzing the turf material. Detail will be presented in later section. And here's highlight. Okay. We are doing solvent extraction for with different polarity in order to cover a full range of chemicals from the turf. We have included lipid and protein in the artificial biofluid to better emulate human saliva, gastric fluid, lung fluid, and sweat. We are also measuring particle size of airborne particle over synthetic turf field with simulated human activity. Task 5 described the biomonitoring program protocol development and personal monitoring protocol development. Chemical identified in Task 4 will be considered in designing the target chemical analysis or analyte in these monitoring protocol development. We are in consultation with experts in the University of California to develop this protocol. Task 6 and 7 are reports, and we anticipate to have an interim report on the study and data by 2018. And also, we will have our risk assessment report by the end of the study around mid-2019. So this is an overview of all our activity in the past, and detail on technical discussion is in the next agenda item. Last slide. This diagram provides an overview of the study timeline. At this time, March 2017, we are in the middle of hazard identification. We are also developing our time activity behavioral pattern study, and we have been very actively doing sample collection and sample analysis, method development, and field selection development. And very soon, we are working on the biomonitoring and personal monitoring protocol development, and at the end, we have our report. So the first 3 tasks, the hazard identification, the time activity behavioral pattern study, and also the chemical analysis of the sample will be discussed in this meeting. Turn back to Dr. Baum.
So thank you, Patty. At this point, I want to ask if any of the panel members have any particular questions or comments. This was an overview. We're going to be going into more detail as the day progresses. Dr. McCone.
I guess just a general comment. I mean, it's encouraging. It appears that the advice of the panel was taken quite seriously, that most of the items here are what we suggested and more. So, and I think the one thing is that, I mean, looking at this, there's might be an expectation that OEHHA will get different results because the European study, it looks like they were using the fluids they were using were probably not the ones we recommended last time, that OEHHA is actually going to be, so they really were not the saliva with lipids or the sweat with lipids and more accurate composition. So there might be an expectation that result is interesting, but maybe this one will be more relevant because of the way it's structured.
Any other comments at this point? Oh, Dr. Kyle.
Yes, thank you. This might be a little bit along the lines of where Tom was going in that, you know, the reason that you all are doing all of this is to answer questions about these materials, right? And so we have a lot of studies and data that are coming out now, and it is a very impressive amount of work, and I thank you all for getting it out to us and everything. But two things that come to mind for me are, how do we explain what was done here that adds on to what we knew before? And what questions are we checking off as we go along through these various analyses? And so you all are understandably presenting this from the point of view of the research. You know, this is what we found, this is what we did, blah, blah, blah. That's good. But there's a step also of, okay, well then we've learned this much more than what we knew before from the literature review, et cetera. And I think that could be brought forward in a way that would help people get more bang for the buck, in a sense, understand more about what we're getting out of these studies. And so part of it is, for example, Tom's point that, well, one thing that you did is use these different fluids, you'd expect to see more, you know. So when we go through and look at these other studies and say, well, they didn't find anything, blah, 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 it doesn't sort of elucidate what they did and didn't do in the different studies, and then what you're doing that might be better or cover some different space. So I'm kind of thinking, well, can we, you know, if we have 4 or 5 major questions that we're answering here, can we help put into perspective, well, what part of that did the EU do? And then what did the people in the Netherlands do? And what is NTP going to do? And what has EPA done? In a way that helps us kind of follow along, you know. Because I, for one, you know, I'm sort of simple-minded sometimes when I look at the stuff. I'm trying to figure out, well, what's the list of chemicals that we have now? And I couldn't really map that across these studies, like, well, and I think we ought to be able to do that, you know, probably somebody can do that, has all those pieces. You could say, well, all the ones that have been tested, you know, these people looked at these 3 and these people looked at these 7, et cetera. So we could kind of see that, et cetera. So I don't want to go on too long about this, but how these data are going to help us know whether we can answer the questions that we have and what questions those are. And then, of course, the next logical thing I won't bring up now is, what are we going to do about the answers? You know, what are the implications of those answers for any action? I think it would just make this more valuable to the part of the audience that's in the public. So thank you.
So might I just try to restate what you said?
Please.
Thank
you.
I think it would be, I agree, it would be helpful for communication with the public if we could identify data gaps and then how the research that you're undertaking tries to close those data gaps. Is that somewhat of a fair restatement?
Very succinct restating, thank you.
Thank you. So
That means I get to go?
yes.
Turn this on. So this is sort of along the same lines, and I know that you guys have been really collaborating strongly with the federal partners, but I think it's really important as you go forward to collaborate and try to standardize things where you can. It reminds me of when we did PM exposure studies in the early 2000s. We had 3 or 4 different organizations doing them, but we standardized it. And what happened is that you were getting the same kinds of results every place. And so if you can start to build up a body of data that is done with similar protocols or as similar as possible, then in fact you begin to develop reliability, sort of an idea that this is what's happening rather than, well, this study had that gap and this was a bit different and that was a bit different, and so we still don't know where we are. And so I really would encourage that, especially as we all know, each one of these studies can only do so much. So I would strongly encourage, further collaboration and really serious collaboration on this if the federal studies go forward.
I would second and third that, but Dr. Abel.
Just
Or
a quick question
Mr. Abel.
on the information on timeline. You mentioned that you would be reviewing sort of the first 3 bars that you showed. Are you going to say anything later in the afternoon about the biomonitoring, personal monitoring plans?
The biomonitoring really depends on what chemical you want to analyze, what chemical we're trying to track. So data coming from the task force is critical. It will develop based on what we find. It's being discussed, the plan is being rolled out, but will be in later stage once we get more data. But we do start planning on it. We don't have any presentation today on it because it's very preliminary.
I want to add that biomonitoring and personal monitoring, in the beginning, there will be some kind of a feasibility study, and then maybe follow up with a protocol development.
Any other panel comments? Dr. McCone?
I mean, to Dr. Sheldon's point, I mean, I do, and I think we'll talk about this more, but the idea of consistency across different studies is going to be problematic if everyone's doing it differently. And I know this has come up with, I mean, going way back, at Lawrence Berkeley Lab with the indoor emissions type studies, where until people figured out how to do it, everyone in the world was getting different results because everyone was using different kinds of chambers, different temperatures, different protocols. And once you get standardized, then everybody's results can be put together, and we get, instead of getting a little chunk here and there, we start getting a coalescing of ideas or results. So.
Well, that was efficiently done.